All right, so I guess we're live right now. Welcome everybody to yet another Journal Club brought to you by Life Extension Advocacy Foundation, LEAF, and Lifespan.io, where we have all of our projects for everybody to take a look at and donate to. My name is Oliver Medvedik, and I'm the director of the Cambar Research Center, Biomedical Research Center here in New York City at the Cooper Union. And today's paper is a paper dealing with Alzheimer's disease, and particularly neurofibrillary tangles. And before I go any further, and before I forget, I was reminded by Keith that, what is it, today is Giving Tuesday, right? Not to be confused with Shrove Tuesday or Fat Tuesday, but Giving Tuesday, right? So that means that I guess now is the official time to donate. So if you've ever felt a guilty pang that you haven't donated to a worthy cause, now is the time to go to lifespan.io and give to our reoccurring donations campaign. If we don't have a campaign up there now, I think David Sinclair's campaign just successfully finished. Do give so we can keep this broadcast going. All right. So without much further ado, I'm going to discuss, let me pull up the paper that we will be discussing. It is, I believe, an open article. So way too many tabs open here. Bear with me. I was looking up all the senolytics that are in use right now, because senolytics is a topic of the paper by Quercetin, Dissonant, a whole bunch of others. Okay, share screen. Here we go. All right. So this is the original paper. Tau protein aggregation is associated with cellular senescence in the brain. Musi et al. And I do believe it's an open journal, so anybody can access it. It is from San Antonio, Texas, at the Barshoff Institute for Longevity and Aging Studies. And in case you don't know, I'm going to stop sharing with what tau is. Just a little bit of background for everybody here. Tau is a microtubule-associated protein. This is something I pulled up just doing some Google foo here. So this is an artist's rendition of a diseased neuron. I don't know if you can see this. Is this clear, Steve? Can we see this picture here? Bunch of little beach balls.
Yeah, it looks all right to me.
Okay, so basically, this is this tube here is an axon, so part of a neuron, and a neuron is packed with these things called microtubules, as all of your cells are. But microtubules have a number of different functions. They're structural elements, but they're also used as highways to kind of conduits to have motor proteins that carry vesicles up and down. So if this is an axon and you have synapses happening, which are not shown here because this is cut off the cross section of an axon, these microtubules will be like little highways, and they need to be stabilized. So all these little balls here are basically microtubule dimers, so alpha beta dimers of tubulin, and they form these tubes. And these tubes basically are dynamic, so they break down and they reform continuously. And tau is a protein that basically is a normally occurring protein that binds to the sides of these microtubules and it stabilizes them, basically. And if you have a defective, if you have a form of tau, which I believe gets aberrantly phosphorylated, it forms these clumps, and these tau proteins, which are under a family of proteins called MAPs, or microtubule-associated proteins, can't do their job. They kind of clump up together, and this causes a destabilization of microtubules, right? So your neurofibrillary tangles are basically tangles, clumps of these tau proteins. So, I'm not sure if the tangles themselves have also a toxic effect, but certainly destabilization of the microtubules would lead to defects in neurons, especially since they need to transport neurotransmitters to the ends of their axons, axonal termini. Sorry. Okay, so that's kind of one of the manifestations of Alzheimer's disease. The other being these other external tangles, amyloid beta, and so basically there's these proteins that clump up. So we won't be talking about amyloid beta. This paper focuses on another pathology of Alzheimer's, which is neurofibrillary tangles. And as they mentioned in the paper, NFTs, which is just the abbreviation of it, these NFTs happen in, I think, dozens of different pathologies that can afflict the brain. So not just Alzheimer's, but brain traumas and a whole bunch of hosts of other brain illnesses where you see this pathology of NFTs happen. Okay, so let's go back to the paper. All right. So what they do in this paper is, and they mention, I think this is the first time, I'm just shuffling notes here to see where I have it highlighted. It's the first report for a role for cellular senescence in Alzheimer's disease-associated neurodegeneration. So basically, they implicate the formation of these tau protein, these NFTs, these tau protein tangles, this aggregation with cells that are in a senescence-like state within the brain. And they look at human brains and they look in mouse models. And they showed that there's this association with not only these NFTs that obviously cause diseases such as degenerative brain diseases, including Alzheimer's disease, and they mentioned progressive supranuclear palsy, et cetera. But they lead to a senescence phenotype, right? So basically the cells become senescent. And we talked about that quite extensively in a number of other journal articles that we've had here, where cells basically, I guess in the brain, they normally wouldn't be dividing, but they would be, what would happen is they would overexpress factors such as P16, which we'll take a look at in a moment, P21, which make certain the cell is arrested, and also the cells start to secrete pro-inflammatory cytokines. That's a bad thing. So that leads to all sorts of toxic effects to surrounding cells. So this is, I believe, the authors state that this is the first time that association has been made, right? So what do they do? Well, they work with mice that, again, this is something that people complain about is, there's mice normally don't get Alzheimer's, so you have to come up with an artificial system to induce these problems in mice. So they use these mice that are these NFT mice, which, so they have tau wild type, and then they have mice that are expressing tau NFT, which they use this RTG tau P301L, blah, 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 4510 transgenic mouse line. It's probably a tau that's, I think, aberrantly phosphorylated that leads to these tangles. Okay, so it's a mouse model, it's the best you can do with the tools you have at your disposal, and and it does exhibit a lot of these hallmarks. They don't, if I would have one gripe with this paper is that they don't look at really, there's no experiments here that look at the motor function of the mice, or the physiology of the mice, or they do a lot of molecular biology here. So again, the point of this paper here is to kind of make that connection between tau and senescence. So figure 1. So a lot of these figures are basically there to make this senescence, make this connection with senescence. And what they do is they use this kind of system here. So I'm just kind of making sure that which ones they use. So they first start off with humans. So they have human brain sections that they are from human Alzheimer's disease neurons. And they basically, what they do is this ingenuity pathway analysis. They basically isolate neurons using this kind of laser microscopy system. That's pretty cool. That you can literally pick off neurons that I guess they have growing on a plate, and that are stained for for tau, and then they basically look at RNA levels. They look at the expression of genes, and they do these correlations. And here are p-values, and these are basically, you'd have to take a look at the supplemental to see how these, what are the specific genes, but the genes cluster with cell survival, cell viability, inflammatory response. So basically the point being NF-kappa B complex. So everything that's associating with you know, everything that's associating with senescence is up-regulated in cells that have been selected that have this tau, you know, this neurofibrillary tangle phenotype. And these are from human cells. So pathways induce upstream, and they, so predicted upstream regulators in B. So that's, so this is from humans. I'm just making sure that I don't cross the streams here. At some point, they're going to switch over to mice. Okay, see, predicted upstream regulator gene transcription in tau NFT mice, and that's their you know, transgenic mouse. And they basically pick neurons, you know, 6 months old versus 2 months old, I believe, after the onset of neurofibrillary tangles. And a lot of TNF, so on and so forth, a lot of these pro-inflammatory markers are up in these mice that are expressing neurofibrillary tangles. They also look at another marker of senescence, which are these H2AX histones, and they look at expression levels and tau wild type, tau NFT. Make sure I've got this straight. So this is control. Tau wild type is this an old one? Old mouse? Let's see. Sorry, this here. Represented immunoblot generated by capillary equivalent bound fractions. Mouse forebrain homogenate probed with anti-gamma H2AX antibody. So this is a histone that's found in as a consequence of DNA damage, which is also correlates with senescent cells. And I believe, so this is a control animal, and I have to go through the text here to see what the control is versus tau wild type. But I think this is an overexpression of tau, and this is tau NFT, which is the toxic variant, and the levels of gamma H2AX go up. So this is total protein. So here's a correlation here going up with mice. And they also look at expression levels of P21, which is, I believe, CDKN2A and CDKN1A is P16, which are tumor suppressors that are found upregulated in senescent cells. And these go up as well, particularly with tau NFT mice. So I'm going to stop sharing there for a moment. So basically, senescence factors go up in cells that have been picked from brains from humans that have Alzheimer's disease, but also in that have neurofibrillary tangles, and also in mice. So this is a correlation. I think this is the first correlation there that's kind of being shown in this paper, where you have a connection between NFTs and senescence. So I think that's the kind of the big take-home message from here. Share the screen back again. Okay. So now to figure 2, neurofibrillary tangles associated with upregulation of SASP gene expression and NF-kappa B activation. So again, this is in mice, I believe. Yeah. So these are from mouse forebrains. So they're, again, looking at kind of in a little bit more detail RNA expression levels of senescent markers that are indicative of senescence. So markers that induce inflammation, and the RNA levels go up. P65 is a component of NF-kappa B, so that's a big component of that's senescent cells. So nuclear P65 levels in tau NFT mice go up. So total P65, so nuclear P65 is, it's a transcriptional factor, so that's where its activity is going to be most present. So nuclear levels go up of P65. And again, nuclear lysate, again, total lysate, P65 levels don't go up, but nuclear lysate, so you have NF-kappa B basically migrating to the nucleus to essentially switch on the senescence phenotype, supposedly. So I'm going to stop sharing here. Any questions from anybody? No, that's a little fast, but they're basically they're trying to make the connections here between, neurofibrillary tangles, or particularly in their mouse model, they had some human cells there as well from Alzheimer's patients, and you know, the senescent cells, senescence phenotypes. And they're not yet looking at any. I don't think they're looking at specific cells yet. So this is just, you know, like a total homogenic of forebrain, but you know, overall the levels seem to correlate with greater senescence.
Yes. So, oh, we have one question.
Yeah.
It's not related to the paper. Mark Millennium, I believe he used to be known as Mark Century. In fact, I know it's Mark Century and is now Mark Millennium. Asks, can you turn your mic up?
My mic? Oh,
yes. You need to eat that mic. Eat that mic.
Well, it's kind of flexible and it sort of moves away. Okay, let me see if I can. Is that better? Is this coming?
It's further away now.
Oh God, it was all right last time. I can't really. Can I turn my mic up? Is that? This is what I get for getting a cheap headset. It was okay last time, wasn't it?
You see, I think mine's a little bit more flexible than yours. See, look. Mine stays there.
Is it clear when I hold it like this?
It's better, yeah. It's better, but you can't really sit there with that. Well, that's it. We're going to have to write to Santa Claus, folks. It sounds like Oliver badly needs a mic, a new mic, and we do need some new equipment. So Giving Tuesday, you know.
Yeah, you know. So, you know, we work on a complete volunteer basis. So, you know, it's not only am I, you know, not only do I run the hair club for men, but I'm also a client, right? So I also donate money. Nobody gets that joke, right? It's an old commercial. But yeah, I buy my own equipment for this stuff. So if anyone wants to get me better headphones. Anyway.
Yes, well, welcome to the wonderful world of 501Cs. And relying on operating things on a shoestring budget, right? So we do our best, but okay. I think we're going to have to write to Santa Claus and have a look, see what he's got in stock.
Okay, so hopefully I'll talk a little louder, and having this thing moved up a little bit will make it better. Okay, so let me go back to the paper, share screen. All right. Okay, so on to figure 3. So here they basically, now they look at mitochondrial function, you know, which is also, you know, defective in senescent cells. They do this, they look at oxygen consumption. They use this SUIT protocol. I'm not going to go over the details of this, but these are basically traces. So these are control traces up here. And these are different substrates, basically, you know, that, you know, for that measure electron transport, and essentially these are, you know, in picomoles per milligram, and these are substrates that are, I guess, you know, some of these agents are used to decouple electron transport, such as FCCP. So levels going down in all of these traces mean that there is kind of more defective mitochondrial function happening, and oxygen flux looks like it goes down. So the red circles here are, again, so these are, you know, oxygen consumption in O2 flux per mass. So per mass of different, I guess they measure the mass or the quantity of, these are components of the electron transport chain. What doesn't go down are the mitochondria. So they look at CS activity, which is citrate synthase, which is basically, I believe it's in the electron, it's in the Krebs cycle. So that enzyme, the amount of it basically just correlates with the amount of mitochondria. So what they're trying to show here is that, you know, there's defective electron transport in mitochondria. The mitochondria are acting aberrantly, but there's still the same amount of mitochondria present. So there's, you know, mitochondrial dysfunction, which also correlates with, you know, senescent cells. So they're basically characterizing these cells, you know, based on if it walks like a duck, quacks like a duck, it's probably a senescent cell, right? So they have all these cells that have neurofibrillary tangles, you know, that are from these tau aberrant mice. You know, all these cells derived from areas that where you're going to have a lot of neurons, such as forebrain. You know, they have senescent phenotypes. And then figure 4, before we get to the last figure, which is really the kind of the punchline, is let's see, figure 4, and then there's table 1, which they actually human postmortem brain characteristics. So, you know, causation doesn't really, you know, it's causation, correlation, right? It's because something's correlates doesn't mean it's causative, but, you know, they throw this in, which is human postmortem brain characteristics, where PSP is that condition. What does it stand for again? I have to, scroll up here, remind myself. It's the progressive supranuclear palsy, right? PSP. So again, so they get brains from, you know, from donors that have passed away. Here's their sex. The MMSC score is basically a score on it's a cognitive test, right? So people that have PSP scored less on it. P-values here. The brain mass seem to be, you know, identical, and what they're showing here is that people that have PSP, the tangles, you know, go higher. So, you know, there's a correlation. Whether it's a causation, that doesn't address it. But what they do here is so then they look, so what they do, I think in figure 4, I'm trying to see if they look at human cells in figure 4 here. So they look at CDK2A, which is a P16, and that was significantly upregulated, they say, in mouse and human brains with NFTs. And let me see, where are the human brains in this? So in here, what they do is now kind of showing that the correlation is actually a causation. They do a genetic ablation of endogenous mouse tau. So here in these tau NFT mice, so MAPT is microtubule-associated protein tau. I'm not sure how they genetically ablate it. To be honest, I have to take a look a little bit more in the paper. I'm assuming they do it conditionally, meaning that these mice haven't been raised to have no tau, because that I would think that would be lethal, but maybe not. Maybe it's maybe they can shut down the expression of tau using a chemical compound, but I have to see how the genetic ablation is actually done. But suffice it to say, if they genetically ablate tau, the amount of P16 expression levels, or CDK and 2A, go down significantly. Brain mass stays about the same. Older mice have greater P16 levels, you know, over a period of time. Significant reduction in brain atrophy. Say significant reduction in brain atrophy. Well, I guess they say that's statistically significant, but okay, kind of a smidge, right? Brain mass goes slightly up. And in D, okay, so here's basically silver staining. So you're looking at neurofibrillary tangles. So PHF1 is, you know, a antibody against. So I guess this would be a silver stain. So this, not sure if that's the same protein, but flipped. This might be a flipped image. So this is, you know, one cell. Okay, so I'm not sure exactly. I mean, okay, so it's staining for neurofibrillary tangles, but I'm not sure exactly what that's supposed to really show me in 18-month-old tau hippocampal neurons. I don't really have a control slide here. And I'm just jumping to I here, which is actually human. So this is a human, this is immunoblot from actually cortical brain homogenates from, these are from the PSP brains that were found in the table below with total tau antibody. And then they show the highest, HD7. So the individual, the highest CDKN2A expression, panel G, where's panel G? So this is from an individual. So these are P16 RNA expression levels. So the one that had the highest level, this kind of outlier here, had highest molecular weight of tau, meaning more neurofibrillary tangles. So, you know, data point of 1, but okay, there's a correlation there. So you have more, you know, for people who perhaps have higher levels of tau NFTs, they probably have much more P16, which might indicate more senescent cells in their neurons. So basically, for humans, you know, I think this paper kind of lays the groundwork to do some more human studies, to take a look at human neurons to see if really there is this nice strong correlation as they showed with this mouse model so far. So again, this is a mouse model. You know, you're overexpressing defective tau protein and you're showing senescence. Whether or not that's a real connection between, you know, all the human conditions where you have aberrant tau and senescence, it's certainly tantalizing that, you know, that might be the case. So what's the big deal, you know, when it comes to treatment options? Well, we now enter into the slide that talks about senolytics, because senolytics are used to basically target senescent cells. So if senescent cells are, you know, if senescence is a problem that, you know, underlies cells that have NFTs, neurofibrillary tangles, and, you know, perhaps Alzheimer's disease in general, and a lot of these other, you know, pathologies of the brain, then perhaps we should be looking at senolytics or things that are also targeting senescent cells. So let me kind of go back to paper here. And really figure 5 is really the punchline. This is where they take a look at 2 senolytics in combination, Dissonant and Quercetin. And I'm just gonna click on, so Dissonant is an approved chemotherapeutic drug. So this is from Wikipedia. Oh yeah, it's Giving Tuesday, so Wikipedia wants donations too. So if you want to donate to Wikipedia, we don't get any kickbacks, but I do use Wikipedia. So I did donate in the past to Wikipedia. So, you know, just FYI. Anyway, Dissonant, this is the chemical compound. It looks quite different from any of the other senolytics that we've talked about before, such as these plant polyphenols. So Quercetin is the one that they use in the paper here, and it's a plant pigment, a flavonoid. So I'm not going to embiggen this, but is this big enough? Can we see that, Steve? This on the screen, Quercetin. It's like a 3-ringed molecule.
It's a little small, but I mean, you know, it's not too bad, I don't think.
Yeah, okay. So I just want to get that point across. So and there's a couple of others. So they use quercetin, but I'll just mention a couple others. This is apigenin, and which may be a senolytic. Steve, I think you got some thoughts on this. But there's also fisetin. Some of you have also heard about fisetin. And gee, I can't find the screen. Oh, here it is. And you'll notice that they all look alike. So they're really, really, really similar. They just, all 3 of those compounds, except for the Dissonant, those plant polyphenols, they're basically just a positioning of hydroxyl groups, which is really the difference. You know,
which sort of ties in with the question that Mark's asked, Mark Millennium. And he's saying senolytics, nutraceuticals or supplements or whatever.
Some of them are. Some of them are.
He says quercetin, fisetin, which is mentioned, piper longum, which I understand is from a pepper or like a chili, and tocotrienols. I can't even say that word. Tocotrienols.
Yeah, and fisetin, for example. I mean, these are plant polyphenols. They're found in plants. I forget how many milligrams per pound you get them in strawberries, right? But
it's a lot. Not a lot. Sorry, not a lot at all.
Yeah, it's a little. It's something like if you wanted to take, like, in these papers, like if you read papers where they use fisetin as an plant extract to give to mice as a senolytic, you'd have to ingest something like on an order of 50 to 70 pounds of strawberries. Like, you know, like, so we're talking about really concentrated dosages here.
Oh yeah, it's like I worked out how many onions it would take for an average 500 milligram human dose of quercetin, which is the usual supplement amount, and it was something like 25, maybe 30 red onions, you know, for the equivalent of a pill. I mean, that's how small the amounts are.
Yeah.
But yeah, to answer Mark's question, was there anything else? Oliver touched on it earlier, apigenin, which is an extract from chamomile, and structurally is very, very similar to quercetin and fisetin. And again, it's another plant polyphenol. And there was a study in 2012 by David Sinclair with quercetin and apigenin, and he demonstrated that it inhibits CD32, which is an inflammatory signal, which is found in SASP. And when they did, levels of NAD were restored. So take it with a pinch of salt. But my thoughts on the matter are, I think apigenin could potentially be a senolytic or a senomorphic. But we'll talk about senomorphics later, perhaps.
Mm-hmm. Yeah. So let's first wrap up Figure 5, which is really the, you know, the punchline of the paper here, where they actually see, okay, so all of these, you know, all these connections that we made in the paper with neurofibrillary tangles and tau and a senescence phenotype, what happens when you actually treat these mice with senolytic or a combination of senolytics? So they use quercetin and dasatinib. And here are basically just, you know, slides. What you're seeing here are, you know, they're basically saying a senolytic treatment reduced neurofibrillary tangle burden, right? So also ventricular enlargement. So basically, so here are slides that look at cells that have been stained for antibodies. So DAPI is just nuclei. Phosphotyrosine is, well, it's a phosphorylated tyrosine, but I think that correlates with the tau protein, and PHF1. I think it's also tau, and I think it's the phosphorylated tau PHF1. Total tyrosine phosphorylation. So here is tau NFT cells with vehicle, which is basically just your control, and so these are 23-year-old tau transgenic mice. I don't know exactly how much, it's somewhere in the paper, how much they fed the mice and for what the protocol was. But basically when they did, you know, histology, immunohistology, brain sections, and they stained, you know, and then they, again, the numbers, I'm not exactly sure what the field of numbers was, but they say that this is statistically relevant. So this is NFTs, you know, over DAPI, so over total amount of cells. So B is basically just a bar graph of all of these, you know, slides kind of, you know, that have been counted. So, you know, you do see less phosphorylated tau, right? So less in tau NFT after DQ treatment. You do see, so these look like MRI cross sections. So these are control animals. These are tau NFT vehicles. So you can see these giant ventricles here. I think those are like spaces in the brain, if I'm not mistaken. And basically this is just, you know, less gray matter in the area here. And you can see with DQ, you know, they are bigger, but you could see that there's a substantial reduction in volume of these ventricles. And you could see that the ventricle volume, again, this is a quantification of ventricle volume, you know, it does go down. With DQ treatment, overall total amount of, so these names here, new ends, PSD95, these are all proteins found in neurons. So so we have synaptophysin and so on and so forth. So what they're basically saying here is that the total amount of, you know, neuronal proteins found in normal neurons goes up. So presumably you're gaining, you know, you're gaining neurons, right? So you're you're improving the animal, you know, in that respect. They do some other studies that are not shown here that are, well, that are shown here, but they're, I think, in the supplemental, which is cerebral blood flow, which is improved. You know, so I have kind of two things to kind of slightly gripe about. One is that I've never worked with these mice, so I don't know exactly how easy or not easy it is to work with them. Does all of this treatment correlate with, you know, behavioral changes that, you know, can the mouse now think like a normal mouse, right? I mean, I don't know how screwed up a tau NFT mouse is, right? So if you make it better, at least, you know, at using immunohistochemistry, does this correlate to a mouse being able to find itself out of a maze a little bit better, right? Is the physiologic responses, are they improved? So I don't know. I think that would be a great follow-up experiment to see if all of these, you know, internal effects can basically be mirrored in the behavioral studies of a mouse. The other thing which maybe somebody could answer for me, and I mentioned this to Steve before, is, you know, and they mentioned this in the paper here, in their discussion. They mentioned, and I'm just going to read right from the paper, and I'm going to just stop sharing here, is that aberrant cell cycle reentry causes neuronal apoptosis and AD-associated pathology, AD being Alzheimer's disease, and requires soluble tau. The observed increase in NFT-associated CDK1A and CDKN2A or alpha, which are P16 and P21 gene expression, may allow so that's their hypothesizing, may allow stressed neurons to abort cell cycle reentry and enter a cellular state similar to cellular senescence. In this way, NFTs formed in early pathogenic stages due to acute stress may initially protect neurons from cell death, but then contribute to neurodegeneration later in life through senescence-like mechanisms by altering the bioenergetic state of the brain, meaning, you know, mitochondrial dysfunction, and upregulating the toxic SASP, or senescence-associated secretory phenotype. So they're kind of suggesting that initially senescence is a good thing because it prevents neuronal cell loss through apoptosis. Yet they're giving senolytics to these mice, which, as the name implies, they cause cells to die, and particularly senescent cells. So if you're getting rid of senescent cells, yet you're now improving brain function by having more neurons, I guess, ostensibly reform and improve brain mass and reduce ventricular volume, then I'm not sure how to correlate that. Because I don't know, I'm just having a logical disconnect here with, you know, like, you know, senolytic good because it promotes neuronal apoptosis, you know, NFT potentially good because it prevents apoptosis. And I'm just not, there seems to be some kind of disconnect here. So either the senolytics here are doing something other than promoting apoptosis of these cells, and they're not ablating these cells, or they are being ablated and the normal neurons are growing back to kind of fill in the gaps, which would be interesting in of itself. I'm not exactly sure. So clearly senolytics, at least the two studied here, are having a beneficial effect. But I'm not sure how to logically wrap my head around that, you know, that causing neurons to apoptose is going to help you, if that indeed is what these senolytics are doing here. I guess the question is, are you better off with a dysfunctional neuron or no neuron at all?
I would probably say that you're better off without it.
And have the other ones do a better job, the surrounding neurons?
Yeah, because let's not underestimate the bystander effect as well in senescent cells. which is that they poison their neighbors. So you can have like one senescent cell and you put it in a bunch of healthy cells, and before long, all the healthy cells adjacent to it become senescent too. They start to give off the same signals. So my gut instinct would be to say here, it would be better to get rid of a neuron that is fully senescent. And let's be honest, that is a gray area in itself.
No pun intended.
But yeah, no gray matter intended. But yeah, it is very much a gray matter in respect of, well, is a cell really senescent or isn't it? You know, because we know that there are different sorts of senescence, or we think there are now different sorts of senescence. So can some neurons be recovered, and are some gone too far? Is that what's possibly happening?
Well, from their data here, it shows that the brain mass isn't decreasing. So that leads me to think that either getting rid of senescent cells promotes neuronal growth, or the senescent cells are being converted to normal cells.
Or there is another option. I'm just going to put this out here. I've touched upon it earlier. Or are the compounds being used not actually senolytic per se, so they're not killing the neurons, but what they're doing is they're masking some of the effects of the secreted cytokines that senescent cells make, which is called the SASP, a collection of like pro-inflammatory signals. It are the compounds, rather than killing the cells directly, reducing in some way or inhibiting or blocking the SASP or modifying it. Are they, rather than senolytic, are they, as some people are now coining it, xenomorphic? So compounds that change the pro-inflammatory profile of senescent cells. So in other words, they're calming down the inflammation rather than actually disposing of the cells. And in doing so, is that allowing these senescent or semi-senescent, if you like, neurons to recover some level of function? Is that possibly what's going on as well? Because we don't know whether they are actually killing those cells. So is it a xenomorphic?
I don't know.
And we know that, I mean, things like apigenin, for example, which you mentioned, and quercetin. David Sinclair showed in 2013, he did a study, and they showed that when they used them, it reduced levels. I said CD32 earlier. Sorry, that was to do with the Sega. It was CD38 that I meant, and that's part of SASP. David showed that when you gave the mice these flavonoids, it reduced pro-inflammatory CD38, which is associated with immune reactions. So I'm putting it out there and saying perhaps what's going on with a flavonoid in particular, quercetin here, is it tamping down that immune overreaction and reducing inflammation? Is that what it's doing, rather than directly killing the neurons? That's what I think might be going on. But that's the best I've got. Victor, have you any thoughts? I mean, that's my best thought, that it's not killing per se, but it's inhibiting inflammation.
Well, I mean, also we don't know if it's xenostatic or just xenomorphic. If it will modulate the SASP or just stop it.
So, oh yes, that was the other thing now. Victor had introduced me to. See, they're inventing all these new things all the time, Oliver. We had senolytics, which directly kill senescent cells. Now you've got xenomorphics, which change the secretions or block the secretions that senescent cells make. And now Victor tells me there's senostatics, which effectively put it in like a limbo.
Yeah, they're. So
I can't keep
up, but still present.
So, you know, is it modifying the SASP rather than killing those senescent cells? Well,
I guess then we don't really have evidence that is clear that it does either. I mean, it's hard to evaluate probably.
yeah, it's hard to tell. I mean, we're not seeing a reduction of brain mass, okay? So that can only mean one of two things, can't it, really? Either the neurons are not being destroyed, or they're being destroyed, but they're being replaced. We do know that neurons, that they can be replaced depending on what, where in the brain they are and what type of neuron. But we don't know. More information needed, more research needed, Oliver?
Of course.
More research needed.
And more human research, more human trials.
But I mean,
Yeah.
of course, Sklenicki, he talks about senostatic interventions, but of course, even simple things like dietary interventions are senostatic in that sense that you see a reduction in the SASP, but you are not actually killing the cells. So,
Yeah. So, it's hard to say really, and it's a blurred line. But xenomorphics seem to be quite predominantly, a lot of them tend to be these plant polyphenols and flavonoids. And as you pointed out earlier, the chemical structure of a number of them are virtually identical. So the interactions are probably going to be very similar. But my bet on apigenin, I think, is a good one. I think apigenin is definitely. I can't believe no one's actually tried it in respect of it being a xenomorphic or senolytic, because it's got all the right sort of signs.
The thing that is hard to determine is, of course, that if you have a senostatic drug, will it perform as well in, for example, lifespan or health improvement as a senolytic? Because it might really affect any measurements of inflammation a lot. But I mean, eventually senescent cells will take over and accumulate, and they are bad. So when will they show up and really be dangerous despite having a senostatic?
Yeah, I mean, as I said earlier, before we started the show, those kind of approaches, they're interesting, but I do see them as a kind of a bit like slapping a band-aid on things. It's a very temporary fix. It's kind of like masking over or papering over the problem. Like I used the analogy earlier, you've knocked a hole in your living room wall and there's a dirty great hole in it, and you go, oh my God, what am I going to do? It's like the equivalent of someone going, oh, I know, I'll put some wallpaper over it. And it looks all right, but there's still a hole there, right? So long term, I think senolytics are probably the best solution. And, from what I've read and on the topic, I think removing those senescent cells rather than trying to compensate for them is potentially the better approach here, which ties us in. We have a question from the audience. Would you like a question from the audience?
Of course.
Quercetin?
Arhar.
Oh dear.
Quercetin.
Quercetin, we would. Okay. Oh dear. So, okay. So we have any thoughts on Ocean Biotechnology's sensolytic platform that induces rapid apoptotic response by using a library of plasmids with a selective promoter of the ICAS9 suicide gene? In other words, what do we think to Ocean's approach, the plasmid-based approach here for senolytics? I mean, I know what I think, but have you guys got any thoughts on that?
Well, I have been following it, I have not been following it in any great detail. I mean, obviously, it depends on the application, right? If you're going to be ablating senescent cells before you put them back into a patient, for kind of an in vitro therapy, then I would think that would make more sense. But then, if you're going to be inducing senolytics into a patient, then having small molecules, assuming they perform, as expected, I mean, that's the easier route to go, right? I mean, that's that to me. So I guess it depends on what the application specifically is, and I would have to take a look at their documentation and see what particular application they have in mind for this protocol of theirs.
Obviously, reducing off-target effect would be a positive outcome.
Yeah.
Okay, yeah. I mean, for my 2 cents, I've been following Ocean's work quite a bit. It being one of the SENS research spin-off companies, I'm very interested in the potential of the system because it reduces the issue of off-target. And the problem is with a lot of these, a lot of senolytics, which appear to be cancer drugs, a lot of the cancer drugs appear to be senolytic. I mean, as in actual true senolytic. The problem is there's such potential for a lot of off-target activity with them. And, you know, I know Unity is going after a modified version of, is it Dissonant or Navitoclax? I can't remember. I think it's Navitoclax, isn't it, Victor? Do you remember?
Yeah, Navitoclax, but it has quite some side effects, of course, too.
Yeah, they're using. I think Unity is using a heavily modified version of Navitoclax as their first candidate. So obviously they modified that drug to try and reduce the amount of off-target that occurs with it. And I mean, let's face it, if it works, it works. Even if it's not a perfect first pass therapy and it doesn't work as well as we'd like, it's still better than nothing. But I do see like Ocean's sort of thing as very much sort of second generation following on. And if they can get it right, I think it really has a huge amount of potential there because you're not having to worry about all the off-target issues that you are with traditional drugs. I mean, not to say that you couldn't design a small molecule to be nearly perfect to do what you want, but it's a lot more work than, you know, if you have a system that can be completely tailored like Ocean's, assuming it works. Assuming it works.
I'm also not really sure about this, how they think about the blood-brain barrier for, you know, get into the brain.
Yeah.
Well, you know, there are ways and means. I mean, at the end of the day, if you do need to get a therapy into your brain, you can. I mean, it's a bit grim, but you can literally go via the nasal passage if you have to. So, you know.
And there's been some studies right now dealing with using ultrasound as to kind of temporarily, you know, make the blood-brain barrier permeable. So that in of itself, I mean, you know, we could maybe have a discussion on kind of combination therapies and seeing how some of these things could actually be delivered to the brain, because I know there's some, yeah, using ultrasound could be one methodology. Now, for this paper here, how did they get the, I'm just looking up for the paper we just discussed, when they delivered the quercetin and the satinib. You guys can go ahead. I'm looking up the administration here. Oh, actually, orally.
Yeah.
Yeah, it was just orally. So I'm assuming it managed to, yeah, cross the blood-brain barrier.
Yeah.
I didn't find that they specified very much how it was done.
Quercetin definitely does. I mean, there are so many studies showing its effect on the brain and how it influences inflammation. Quite a lot of people think that quercetin is an antioxidant, and it's ironically it's actually not. It's quercetin is actually a pro-oxidant, and which in a long way becomes an antioxidant, which sounds a bit strange, but what it does is it gives the cell a low level or hormetic sort of level of stress, and then what happens is the cell then responds with its more robustly with its anti-inflammatory anti-inflammatory and such like mechanisms. So effectively, by being a pro-oxidant but a very weak one, it actually antagonizes an antioxidant response from the cells. But there's loads of studies showing its effect on the brain, so we know that quercetin definitely does go through the BBB. And yes, I'm going to call it the BBB. So there we are. So yeah, I mean, you know, plant polyphenols, they're interesting, but again, they are effectively a drug. I mean, you know, let's make no bones about this. What are supplements? Well, effectively, supplements are unregulated drugs, but they are because they can interact with your body just the same as drugs can. It's, you know, so, you know, should you begin testing? I don't know about that. I think caution is advised.
Mm-hmm.
Yeah.
But you know, a lot of people do take plant polyphenols, and of course, people who already have a high fiber and mostly vegetarian diet, they're already going to be getting a lot of these polyphenols anyway. And we know that there is some correlation with lifespan and sort of more plant-based diets. So, you know.
But I mean, that's of course has no, it's not a senolytic effect. I mean, there are so many other factors.
Well,
yeah, and anti-inflammatory.
That's the issue, is right. There's so many other factors. It's really hard to pick them all apart. A lot of these compounds probably have cross reactivities. So, you know, I think you know, the issue is here structuring tests, structuring trials, you know, clinical trials, and that's something that we at Leaf have been talking about doing, is putting together some sort of crowd-sourced, you know, clinical trial using, you know, implementing things that are GRAS, generally recognized as safe, lifestyle interventions, things that you might find in this protocol that some of you probably heard of, called the Bredesen protocol.
But Oliver, I guess you read the GRD list sometimes?
GRD list?
Yeah.
No.
Because there are so many people there who are like older and they take all this analytic and report their anecdotal results.
Yeah, there's a lot of anecdotal stuff out there. So, you know, getting things together and seeing if we can actually robustly like measure the effects. Now, I'm of the opinion that, and again, I mean, we have to really kind of sit down and make sure that we do this properly, right? But I think there is a way to kind of do a multi kind of factorial study and engage the effects without kind of being overly reductionist, right? Because it's because it like for me, for example, if I take 10 things and only one thing out of those 10 things is going to help me, but the other 9 are neutral or have somewhat positive effects, then I'm willing to take a combination of 10, right, to see a response. I don't really need to, and then you can trim off the fluff later, right? If you so to speak, so if you set up a trial in that in a proper way, you could control for kind of many factors all at once. And in a sense, it's also sort of how you phrase it, right? Because, you know, you have a lot of clinical trials that look at the efficacy of a vegetarian diet, for example. I mean, a carrot has hundreds, if not thousands, of secondary metabolites, right? So it's like you don't set up 1,000 different clinical trials for every single compound that's inside of a carrot, right? You basically say, okay, we're just gonna mathematically bin it as one and say this is a carrot and it has everything in there. And is a carrot good for you versus not eating a carrot, right? Just because that's just the way you have to structure it, right? So I think there's a way around to kind of structuring these multifactorial trials where you can combine a lot of interventions. Some of them might not work, but the and we did cover a paper that kind of shed some light on this with, you know, some compounds which sort of kind of started to broach the subject of how to properly set up multifactorial trials, right? So using different chemical compounds. And as long as there's no negative synergy, right? Or I don't know what that would be called, energy between compounds, right? So as long as you don't have compounds that combine together and form something toxic, which is highly unlikely in a lot of these, in a lot of these cases, rice notwithstanding, then you should be okay, right? Because you're looking for something that either synergizes, which is good, or has no effect, which is fine, if it's something cheap, right? Or something that has a positive effect, right? So a lot of these clinical trials that are done now for individual compounds, they have to be done individually because the price of these individual compounds, these pharmaceuticals made by a manufacturer, antibodies, for example, let's face it, due to market reasons, not because of manufacturing reasons. I mean, antibodies should be dirt cheap, but they're not. These compounds are incredibly expensive. So nobody's going to give a mixture of 10 different antibodies, with the hope that only one works, when each of them is being marketed as costing $250,000 per year, right? Then that just doesn't make any sense. But if you're talking about generally recognized as safe metabolites and plant compounds that are already being sold, pennies per gram or dollars per gram, then having a multi kind of factorial intervention study makes a lot of sense, at least to me. As long as we're fairly confident, and I think we are, based on the data that's out there, that some of these combinations won't have any added toxicity, right?
But I mean, obviously, if you are having a trial with senolytic health purposes, you will need to quantify senescent cells, and that would be invasive. That would already be very problematic.
Well, okay. So the question is, what is going to be your endpoint, right? And I would argue to just jump over. So I'll make the argument to kind of pole vault over any kind of cellular or molecular measurements, right? So
physiology.
Basically physiology, right? So I would make the argument that for me, a physiologic endpoint, such as being able to climb stairs or being able to score higher on a cognitive assessment exam, or better respirometry measurements, how much you can exhale, all of those things are really the true endpoints, right? I mean, to me, okay, as a molecular biologist, I want to know how, the epigenome changes. But, do I want to feel better, or do I want to have my epigenome look younger, right? Obviously, you're expecting a correlation between the two. But for me, the more important endpoint are the physiologic responses, right? I mean, in the end, if your epigenome looks fantastic, if all your senescent cells are ablated and you just feel awful, and then what exactly are you curing, or what exactly what's your therapy really doing? So, I think, I would do it the other way. First of all, those physiologic responses are easier to gather, and then once you find beneficial physiologic responses, I think you can then go back and make those correlations, and say, okay, now that we have something that seems to be working really well, is it reflected now in less senescent cells or less senescence associated secretory phenotype? Is there an association with a younger looking epigenome? Is the methylation status improved? Then you could do all of that. But I think, starting the other way, highly reductionist, and then, getting to a point where now let's see if it makes you feel better. I think that's backwards and will slow things down considerably. That's not to say that you might miss a few things, but I think a trial could be constructed, and that's something that we've been talking about and doing behind the scenes, bit by bit, where we could see what's efficacious as far as what really matters to most people, which is my physiology improved, right? Is my blood pressure go down? Do I feel less joint pain? Am I able to, is my aerobic response better, improved? Can I run? Is my endurance improved? Is my memory improved? I mean, that's really what everybody's interested in the end. And obviously, there's going to be a correlation between the molecular and the macro scale. But I think, you know, putting together a study where we look at the macro scale first, you know, I think that's the way to go for a crowd-sourced type of trial, because there's going to be less invasive, it's going to be less hurdles for participants, and we can get a lot more data much more quickly, and you know, with also less regulatory hurdles. You don't need to draw blood. You know, you can do that later, and you should do that later. But I think doing it in this order would be better. You know, for this type of trial that we're thinking about.
But obviously, we'll need like a doctor who objectively quantifies things.
Well, we need some people. We need people that can put together the infrastructure, right? Because obviously, you need to have the data collection. But
because you know it's highly problematic with all this anecdotal subjective evidence.
Of course. So if there's a way to do it, and have a way to do it where we can engage people, where they can, you know, upload their own data. And if we can put the right framework in place that can capture this data in real time, then yeah. So, I mean, the anecdotal data, the anecdotal stuff is out there. I mean, the bottom line is people are doing all of these things anyway, right? They're taking high doses, low doses. They're doing this, they're doing that. They're taking plant polyphenols. They're self-experimenting. You know, there's all of this stuff happening where, you know, a lot of this stuff ends up not being really useful because there's no data being collected, right? It's all anecdotal. You know, self-experimenting is fine, but if you really want to progress the whole field, you need to pool all of that information with all your cohorts, and see if it's you know, really working. And I think getting the data, getting the physiologic data, you know, that I think that would be primary, and getting the molecular data, you know, that would be secondary. You know, that's the way I see it. Anyway, so interesting paper. Very, I mean, it's this is supposedly the, you know, the first and fairly late paper, right? August 2018, where they first make a correlation between senescent cells and tau protein aggregation. So, you know, there's a lot of stuff that's you know, that's being discovered. Many new discoveries being made where, you know, you think people have pinned this down a long time ago, but you know, but not really. No, evidently this is the first such example that's been published, the first connection, and hopefully more such connections will be made with human brains.
Well, it's turning out to be the case that senescent cells are implicated in, well, it seems to be more and more conditions all the time. You know, osteoarthritis, heart disease. They literally are intrinsically involved in anything, which is probably no surprise, as most age-related diseases do tend to have an inflammatory element, like chronic inflammatory. And I'm just going to do a blatant shout out to we have a bit of a celebrity with us today, Mike Bonkowski from Sinclair Lab. So whoop, whoop. There we are. Shout out to Mike, who's joined us. He said his ears were burning, so he must have heard me mention. He must have heard me mention the Sinclair Lab earlier in respect of David's work with apigenin. So he seems to have like a spider sense.
I see.
So I don't know. Is this a thing among scientists? Do they have like science sense?
I don't think so. If they did, then they wouldn't be getting scooped so much, right?
Oh dear. So yes, I thought it was an interesting paper anyway. And again, it just shows you how intrinsically involved senescent cells are, you know, and how important they are. You know, there are a lot of potential issues with it. Obviously, the mouse model aside, I think we've just got to get on really and start testing these things, because we're never really going to truly know, are we? You know, if they do or don't work until we actually try. I think we've reached the point really now. We've gone as far as we can with mice, and I think we're just going to have to sort of take that step forward. I'm pleased to see that obviously companies like Unity are already in human trials, not for Alzheimer's, but they are already testing senescent cell removal anyway. So I'm sort of confident that we might, cautiously confident, is that a thing? That we might actually have some senolytic human data in the not too distant future. And I'm going to stay optimistic about it meanwhile, and, you know, hope for the best, I suppose.
And really that's what, you know, anti-aging and longevity research is all about, right? These compounds that are being tested, whatever indication they're being tested or initially approved for, like Unity, the fact that they're hitting processes that are fundamental to, you know, that underlie pretty much every chronic disease state in our bodies means that, you know, these drugs will be applicable in a very broad spectrum for many, many, many ailments. So just because they're testing a senolytic for macular degeneration doesn't mean that it's only good for macular degeneration. It's going to be good for, as this paper shows, probably a whole host of ailments that are disease related. And that to me, that is the fundamental difference between, you know, the industry that is now kind of in its adolescence, or I guess just hitting maturity or hitting its stride, versus you know, the pharmaceutical industry as it's been practiced so far. I mean, these compounds will not be, you know, just to get around FDA hurdles, they have to be marketed for a specific indication. But just the way the biology works, these are not going to be good for just one specific illness. They're going to be, you know, let's not mince any words. They're going to be anti-aging compounds, and they're going to hopefully each compound will be able to target many, 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 many ailments all at once, which is radically different from how pharmaceuticals have been marketed and tested up to this point.
And just for reference, folks, that's anti-aging in a real sense, not the L'Oreal sense.
Yeah.
So, you know, this stuff has the potential for actually, you know, reversing or, you know, ameliorating age-related decline. So, you know, in a very real sense. So yes, unfortunately, the cosmetic industry stole anti-aging from us, and it's got a bit of a stigma, hasn't it?
Well,
it's not a disease anyone can use the claim, right?
Yeah, which is good and bad, right? I mean, you could, but you know, whatever.
Although slight side tangent, I have actually seen in the cosmetic industry there are topical creams that have quercetin as an active ingredient, and I have seen a study where they use topical quercetin and applied just to the skin, and it did seem to improve like skin elasticity and things like that. And also, interestingly, earlier this year they found out that senolytics also influence things like liver spots, you know, like the discoloration of your skin. They found, again, on testing skin cells, a study showed that it seemed to sort of reduce, you know, liver spots and things, which again is very interesting, because that's, you know, another aspect of aging. It's all to do with lysosens, isn't it, and that side of things. So it just goes to show you how entrenched senescent cells are in all of these processes.
So basically, the suggestion is that we should go soak in a giant strawberry smoothie.
Well, I mean, actually, that would probably cause other effects. I mean, many vitamin C, for example. I mean, that's a tyrosine kinase inhibitor, and that's how it has its effect actually on the skin. So, I mean, yeah, but you don't know where the effect come from, in a sense.
You don't want too much though, because you could go orange, which is apparently a real thing. I've never actually seen it, but I'm told that if you do have too much vitamin C, you can potentially become very orange.
Well, I don't know if it's vitamin C. It's definitely beta carotene.
Oh, that's it. Yeah.
Yeah, because it's fat soluble, and yeah. So if you drink a lot of carrot juice,
that's it. It was carrots. That's what I was thinking of vitamin C.
so you end up looking like a giant carrot.
You become very orange. Like fake tan orange. It would be. I've never seen it, but I think you'd have to have a lot of carrot. You'd have to have a lot of carrot to do it.
Yeah, I think our current U.S. president drinks a lot of carrot juice. Possibly.
The only person I've ever seen eating a lot of carrots is our friend Mike Lusgarden, of course. And he recently did a live challenge on YouTube where he ate, I think it was a pound of raw carrots, and he tried to beat the record. And I think it was both fascinating and frightening in equal measure to see him destroy those carrots. But he beat his personal best. I can't remember the exact time on the clock, but it was impressive. I've never seen anybody put raw carrots away that quick.
Okay, well,
kind of like a wood chipper. But I better warn him. If he does that again, he might be going orange.
Okay, well,
but there we are.
With that, I guess that wraps up today's Journal Club. We will have no doubt another exciting journal article for everybody next month. We'll have to schedule it appropriately because of the holiday season, Christmas time. So we don't want to do it on the 24th and 25th, probably.
No. We have a holiday. So generally speaking, we'll be wrapping things up around about the 22nd and coming back on the 2nd. So even we have a bit of a rest over Christmas. Well, we will be working, but just not doing the regular broadcasting and scheduling. So I suspect it might be a bit earlier in December, to be honest. So we can all at least try and have a bit of a break.
Yeah.
So,
Okay,
but what we'll talk about next, who knows? Okay, so have some strawberry smoothies and some carrot extract with your eggnog, and make it a healthy holiday season. So take care, everybody. Early to be talking about that.
No, they're already selling Christmas trees outside.
So, well, I know what they're doing, but it is. Oh, I don't know. I'm not wishing anybody happy holidays or Christmas or anything at this point. I'm getting. My wife's definitely saying no. It's not. You can't even talk about Christmas now for the, until what day are we today? 27th. No, sorry, folks. Another 4 days, and then we're okay-ish.
Okay.
So we'll have to wish them a Merry Christmas next time.
Okay. Merry post-Thanksgiving.
Merry post. That's it. Merry post. That's not a thing. Merry post Turkey Day, everybody. And obviously, hopefully you'll join us next time if you enjoyed the show, including our silly jokes at the end, which we always do. Maybe consider becoming a patron, one of our Lifespan heroes. And you can check that out at lifespan.io/hero. And thanks for joining us. And we'll see you next time.